Hi AP Bio, welcome to another virtual AP Bio lab. Today we're going to be doing the photosynthesis lab for AP Bio. So the materials that we need to perform this lab include spinach, live spinach. We need syringes that do not have a needle on it, so just a plastic syringe. And then we need 2 cups or 2 beakers. And then in these cups and beakers, one is filled just with water and a tiny drop of a soap solution. And then the other is filled with water, that tiny drop of soap solution, as well as sodium bicarbonate, or in this case, baking soda. So the one with sodium bicarbonate is labeled with CO2. The one without any sodium bicarbonate is labeled without CO2. So I already did this, but we needed to hole punch 10 discs out of a leaf of spinach. So I have 10 for my without CO2 and I have 10 for my with CO2. Now what I'm going to have to do next, and this is one of the trickier parts of this lab, is I need to put the discs into the syringe. So I'm going to remove the plunger. I'm going to put all 10 discs in. So we're going to start by doing the without CO2. So I put all 10 discs in, and then what I'm going to end up doing is taking up some of the water from my without CO2 beaker, and then I'm basically going to create a vacuum using the plunger to remove any gases from the spinach leaves. I'm going to show you an image and explain why we need to do that. Here's a diagram of a cross-section of a leaf, specifically if we're thinking of a spinach leaf, but this applies to all leaves. So if you look here, this again is a cross-section. We can see there's a lot of air space. So this air is either oxygen or carbon dioxide, and it's located in this spongy mesophyll layer. So on the inside of that layer of the leaf. And so the reason we have to do the vacuum is to remove all of that air, remove all the oxygen and remove all the CO2. So there's no more gas left in the leaf. And instead what ends up getting inserted is whatever liquid we were using. So with the without CO2, it's just water that's getting inserted. And the with CO2 beaker ends up inserting sodium bicarbonate, which is an alternative source of carbon dioxide. So then as photosynthesis can occur, or if photosynthesis is occurring in those leaves, we would start to see a buildup of oxygen in those air spaces again. So that is what would cause the leaf disc to float. So if it's floating, photosynthesis is happening. If it's not floating, there is no photosynthesis occurring in that leaf disc. Okay, so now that we know why we need to remove the gas from the spinach leaves, we're going to go ahead and do that. So how that works, again, I'm putting all 10 of my discs into my syringe. So right now it's just an empty syringe with now 10 spinach discs. So there's no liquid just yet. It's just the discs inside my syringe. So I'm going to tap them down to make sure they don't get crushed. Tap them down so they're at the bottom. And then I'm going to put the plunger back into my syringe. And then what I'm going to do, I don't want to crush the leaves. I want to make sure they stay intact. And then I'm going to pull up just a little bit of for this one, without CO2 liquid. So I'm just going to pull up maybe about 2 millimeters, 3 millimeters of this solution. So I'm going to go ahead and do that. So here we go, pulling up about 2 millimeters of the water. Then I'm going to tap, tap, tap to make sure all the discs are suspended. So you'll see right now they're floating. That's because they still have the gas in it. So now what I'm going to do, I'm going to push up to release any extra air. Make sure it's just liquid now. So all the air is released. Then what I need to do is create a seal using my fingers. So I'm going to use my thumb Need to push real hard, make sure there's a good seal, and then I'm going to pull down on the plunger. This is going to create a vacuum that's going to suck out all of the air from the spinach. So we're going to do it a couple times until all of the spinach leaves sink to the bottom. So I went ahead and did it once. We can already see some of them are starting to sink. So they lost the gas, they became more dense. We do still have 2 or so that need to sink, so I'll do it again. Again, I put my finger on, pull down the plunger, hold it, and release. And a tap to see if they'll all settle. And then there we go. So now we see all of the spinach leaves have settled to the bottom. So all of the gas has been removed. That means these ones are good to go. I will empty these into the without CO2 jar. So they go in the same one. And then we're going to repeat this same step for the with CO2. So you can see all of my spinach discs are at the bottom of my beaker, my without CO2 beaker. Okay, so now I'm repeating that vacuum step with my with CO2 jar. So this time, again, I have 10 new discs of spinach, and this time I put liquid from the with CO2 beaker in here. And again, we need to vacuum out all the air. We'll give it a little tap, see if any of them fall. Looks like most of them did. I'm going to do it one more time. You want to be careful not to be doing this vacuum technique too many times, as it can begin to damage the spinach. So about 2 times should be good if you're doing it correctly. So there we go. All my discs are now sunken. We removed all the gas. So these are ready now to go back into the with CO2 beaker. So I now have my sunken spinach discs into the with CO2 beaker. I have the same thing in my without CO2 beaker. So now what I'm going to do, I'm going to place them under a light to stimulate what would be sunlight. And then what we're going to do is watch the time and see and count how many discs float to the top. So there we go, time. Again, every 30 seconds we're recording for each one how many discs are floating. That's 30 seconds. None and none. Here we are at 60 seconds. Still none and none. I will give a light tap to either side to make sure none are stuck. Good. Here we are at 1.5 minutes. Still none floating. Two minutes. 2.5 minutes. Three minutes. Three and a half minutes. Give them another little swirl. Here we are now at 4 minutes. Now I have one floating in the with CO2 jar. Four and a half minutes. There are now 2 floating in the with CO2 jar. Five minutes. There are 3 floating in the with CO2 jar. I give another little tap, make sure none are stuck on the bottom. Here we are, 5.5 minutes. 3 floating with CO2, 0 floating without CO2. We are at 6 minutes. 3 floating with CO2, none floating without CO2. Give them another good tap. Make sure none are stuck. That is 6.5 minutes. We now have, I believe it's 1, 2, 3, 4, 5 floating in the with CO2, none in the without. You're now at 7 minutes. There are 7 floating in with CO2, none floating in without CO2. We are at 7.5 minutes. It's still 7 and 0. Give them another tap. Here we are at 8 minutes. It is now 8 floating for with CO2, none floating for without CO2. Eight and a half minutes, still at 8 and 0. Nine minutes, still 8 and 0. We'll give them another shake, again a light one, just to make sure everything is not stuck. We're now at 9.5 minutes, still with 8 floating for CO2, none floating without CO2. We are at 10 minutes, 9 floating for with CO2, none for without. Ten and a half, 9 floating, 0 floating. Give them another little move. Nothing is stuck. "11 minutes, we are at 9 and 0. "11 and a half, still at 9 and 0. "12 minutes, 9 and 0. "12 and a half minutes, still at 9 and 0. Give them another little movement, nothing's stuck. We're at 13 minutes. We are at 9 and 0. Thirteen and a half. We are at 9 and 0. One little stir. "14 minutes, 9 and 0. "14 and a half, 9 and 0. Here we are at 15 minutes. All 10 are now floating in CO2, with none floating in without CO2. We'll go ahead and take a look at what it looks like from the top as well. So here they are again from a different view. We can again see all 10 are still at the bottom for without CO2, and all 10, after 15 minutes, made it to the top for with CO2.